So, as it's been introduced, I will be talking today about how we looked at sleep-wake regulation in mice using a novel mutant mouse line, which has a mutation in synaptobrevin 2, and how I combined the data I got from this mouse with some computational modeling approach to try and get additional insights. So I've been working in Oxford with Vlad Biezowski, Stuart Pearson, and also Pat Mullen, who is based in MRC Harwell. So I'm not going to introduce too much the two-process model again, because we've seen it quite a few times since between yesterday and today. But my point is these two main processes that are assumed to regulate sleep, so the homozygote process, or process S, and the circadian process, or process C, we don't know quite as much for both of them. So about the circadian process, we know quite a lot about neuronal networks, molecular mechanisms. The homozygote process, we still understand fairly little about the mechanisms underpinning S. And so one approach to try and look at this is what we call forward genetics, which is an unbiased approach in the sense that you don't make assumptions of which genes could be underlying this process S. You just induce mutations or use your QTL approach and then see what genes could have an impact on this. So there's been relatively few studies looking at this. One is a QTL approach by Paul Franken in 2001. And another one is a more recent paper we mentioned earlier, the 6-3 mutant. So that's the Funato et al. Paper, which screened lots of mice using EEG methods to try and see investigate what genes could be underlying differences in sleep regulation. And the mice I'm going to be talking about, so the Sleepy 6 mouse line, was obtained through used a forward genetics approach, and they were initially found to have a reduced amount of sleep, and subsequently the mutation was identified, and it's a mutation in the synaptobrevin 2 or VAM2 gene. So it's a point mutation which leads to one amino acid change in the transmembrane domain of the protein. And as you can see on this little diagram, VAM2 is very important for exocytosis or vesicular fusion. So it's part of the snare complex where it interacts with SNAP25 and syntaxin 1A to lead to this vesicular fusion. So VAM2 is widely expressed across the brain and throughout the body as well. And there's another VAMP protein, VAMP1, but which can't replace the function of VAM2, at least not as far as we are aware. And so these mice are amazingly healthy compared. So if you knock down VAMP2, the mice die straight after birth. But our mice, we couldn't find any particular differences in lifespan, and they had. So a PhD student, before I started my PhD, had done some characterization of these mice. So when I started, I knew that they had an array of mild phenotypes, such as hyperactivity. They don't marble. They don't bury the marbles in the test, for instance. They don't build nests. They sleep in a very unusual posture on their side rather than curling up. So they have these few phenotypes, but overall, they're really doing well. And so what I'm going to present today is all the electrophysiology recordings I've been doing in those mice. I did some molecular and behavior work, but for the sake of time, I will not be presenting that today. So I recorded the EEG in the M1 cortex and visual area cortex, the V1 as well here. And we also implanted silicone probe, so a laminar probe in M1, but on the left, to try and have an idea of the multi-unit activity and local field potentials across the layers of the cortex. So it's, yes, these 16 channels that are here. And what I found is that they have a reduced amount of non-REM sleep, so Sleepy 6 homozygotes, because they're homozygote for the mutation that I mentioned. They have a reduced amount of REM sleep, and then obviously because they have reduced amount of both of the other states, they have an increased amount of wake. But I actually had my PhD viva on Monday this week, and in the discussion that we had, we wondered whether we could actually talk about non-REM sleep and REM sleep. You'll see why, because I'll be showing some raw data a bit later on. But for choice of other words, I'll be mentioning non-REM and REM sleep today, and we can discuss later whether that is actually sleep. So now if you look a bit more at the Vigilant state architecture across the light-dark cycle on a baseline day, what you can see is actually they do sleep quite, so in red are the homozygotes, the mutants. They do sleep quite a lot towards the end of the light phase, and significantly more than their wild-type littermates. But then throughout the dark phase, they sleep much less, which accounts for the overall reduced amount of non-REM sleep. For REM, they overall have less REM throughout the 24-hour days, and then wake is fairly opposite to what you observe in non-REM sleep. But then I wondered, they have less of this sleep or silent state. How does that reflect, how can the bout duration of these different states be impacted? And what you find, which is quite interesting, is here is the absolute number of episodes of non-REM sleep or REM sleep in the light phase or the dark phase. And what you see is actually the homozygotes, they have overall less non-REM sleep, but they have a significantly higher amount of very long bouts and much less of the very short bouts. And you see a similar pattern in REM, although it doesn't come back as significant. So they sleep less, but when they enter sleep, they stay for quite a consolidated period of time in that state. So it's not as if they're constantly changing from one state to the next, and which is quite a nice finding. So now if we go to the spectrum, you have a very dramatic change in the power spectra across most frequencies, except the very slow frequencies, around 0.5 hertz. And this is observable in all states and in the frontal derivation as well. If you look at the LFP, you have the same. And that was extremely striking. You don't see that very often. And again, I will be showing some raw signals, and this decrease in power is actually not too surprising when you look at that. But just before I go into that, this is when we assessed the response to sleep deprivation in these mice. So here you have slow wave activity, so the delta power in non-REM sleep across a baseline day. Then we sleep deprive the animals for 6 hours by providing them with new objects, and then we let them sleep for the rest of the light phase and the dark phase. And what you can see is that, these levels are all normalized against the baseline. So what you see is the wild types have a nice increase following sleep deprivation, and the homozygotes, they do have a slight increase, but not quite as strong. And if you look now into the power spectra a bit more, so here I'm looking at this blue line is the wild types for the 2 hours following sleep deprivation, normalized against a baseline, and then this is the 2 hours afterwards and 2 hours again. So you see the 6 hours after sleep deprivation. But the first 2 hours, you can see you have this very nice increase in slow wave activity and delta power, and you don't see such a big increase in the homozygote mice. So the response to sleep deprivation seems to be quite different as well. But here again, I'm going to go into the raw data because this can be discussed. So here you have these frontal, occipital, and then frontocerebellum EEG in a wild-type mouse. You have the EMG, which is flat, that you can hardly see here because, yes, the mouse is asleep. Here you can see 4 LFP channels from the laminar probe and the 4 corresponding multi-unit activity channels. And what you can see is this nice, sometimes slow waves that tends to correlate with periods of silence that happen across the multi-unit activity channels. And if you go into homozygote mouse, this is what you see. And I promise the scale is the same. I haven't stretched my graph. And you have, so this slow wave, and then it just stays flat for almost 3 seconds. And if you look at the multi-unit activity, there's almost no firing happening here. So it is extremely striking. It is more reminiscent of, I don't know, comatose anesthesia, death possibly. So that's why I've been told this is not sleep. Yes, so we can discuss that later. But this is extremely striking. And again, because you have so little oscillations, that can also account for the reduced power that you see in the spectra. But I had also adapted an elaborated version of the two-process model that had been developed in humans that allow you to look at changes in slow wave activity levels across changes in vigilant states. And I adapted it in rodents, so that is done in black 6 mice. But the point I want to make here is because this model was working well, I adapted it to the Sleepy 6 data. And what you find is the increase of process S, so that's the rising constant of S, is not significantly different across my mice. Well, it's quite variable, so you can argue that maybe it would be with higher numbers. But then if you look at the decrease of how fast this decreases, it's actually much slower in the homozygote mice. And if you look at the upper asymptote, so how high it would go should it continue to increase, again, it's much lower in the homozygotes. This is not too surprising given their reduced slow wave activity level. This was more unexpected because they show, so the slower decay rate, but they show less sleep, which you could think maybe they sleep less because they dissipate the sleep pressure more quickly. Actually, they dissipate it not as quickly. So this question remains open, but that could also be because looking at slow wave activity in a mouse with so little oscillations anyway, during a state that is still debatable as being sleep or not, the conclusions are not so easily drawn. So just to conclude, we can see striking difference in the electrophysiological signals, so decreased EEG power, longer neuronal off periods. The EEG and LFP trace in non-REM sleep is more reminiscent of anesthesia. And the switching between vigilant states does not occur as often in Sleepy 6 homozygotes. So I've shown how these long bouts are here, occur in non-REM sleep and REM sleep. I have more data that support the fact that, yes, the switching between vigilant states is not occurring as easily. And so the conclusion is these mice that we used as a model to look at sleep homeostasis regulation might actually be a better model to look at state switching or anesthesia rather than sleep homeostasis. And I would like to thank my supervisors and the people in the group, those in Boulder, those who have helped me throughout my experiments. Thank you.
Thank you, Mathilde, for showing us some very dramatic results. Are there any questions?
Yeah, I was not completely sure what's the mechanism of the mutation. So is it an hypomorph, or what is it doing?
So
because you said that the knockouts are lethal.
Yes.
So
yes.
the heterozygous nulls are normal, or is it an haploinsufficiency?
No, homozygotes of the knockouts of VAM2 are lethal, but here we have a line with a point mutation, so one amino acid change in the transmembrane domain. And what seems to be...
The heterozygous of the knockout.
Oh, the heterozygous of the knockout? I'm not...
Are they totally normal, or
I think they are, but I can't be entirely sure. It's 2001 short paper. I would have to check for the heterozygous of the...
I was just wondering if it's just a simple dosage effect of the, I mean, that if it's an hypomorph, it's basically the mutation makes the protein a bit less effective, but that's what. But
in the heterozygous for our mutation, they are quite similar to the wild types in everything that we have looked at. For instance, the homozygotes are smaller, have all these array of phenotypes, and the heterozygotes, they really look like the wild types. But in our case, for the, yes, yeah.
Could be interesting to understand what that mutation is doing, but I don't know enough about.
Yeah, what seems to be happening in this case is the vesicle seems to dock, but the fusion doesn't happen as readily. So if you have a single spike, the fusion doesn't happen quite as well. There's a tenfold decrease in fusions. And if you have a train of spike, it just works fine. So it can fuse. It is functional, but not fully functional.
Okay, thank you.
We have time for another question. Can the next speaker start setting up the...
Thank you for your talk. It was very interesting. Just a quick question. Did you see any changes in sensitivity to anesthesia in the Sleepy 6, such as in surgical maintenance or recovery?
Yes. So there were some dramatic changes. All I have is when I was doing the surgery, so it's isoflurane anesthesia. And so the induction didn't seem to be particularly different. They do take a little while to lose the tail pinch reflex, for instance. But the most dramatic change is after surgery. It takes about 4 hours for them to be walking around normally, whereas the wild types, it was in 20 minutes they're walking around again after the surgery. So they take a long time to really recover from the anesthesia, yes.
While he's setting up, we have another question.
A frivolous question. Did you pass your Viva?
Yes, I did.
We have one more question.
You showed EEG that you said superficially resembled anesthesia.
Mm-hmm.
That resembles a deep level of anesthesia, so-called burst suppression. And
Yep.
under those situations, an animal will be absolutely unresponsive. You could lift it up and turn it over, and it wouldn't wake up.
Mm-hmm.
I assume your animals were not in that state.
No, so if you, for instance, during the sleep deprivation, at the end of the 6 hours, they can fall asleep. You can wake them up.
Yes.
But
I think it really is not like anesthesia in any sense.
Okay. Yes. Then. Yep, thank you very much.